Hello everyone, I'm Aybuge and I'm a PhD student at Max Planck Institute for Molecular Genetics in Berlin, Germany. I'll be telling you about my PhD project titled as Annotating Single-Cell Toxic Pseudobulk Clusters. I will start telling you about standard single-cell RNA-seq analysis pipeline to annotate single-cell clusters, and then we'll continue with our efforts to annotate clusters of single-cell toxic. In this roadmap, you will start with your single-cell RNA-seq data and do some sort of filtering, followed by clustering. The primary step when annotating a cluster is to use no cell type specific marker gene expression. So you basically check the expression levels of your marker gene for each individual cell and annotate those clusters showing the higher expression of the given marker gene as per the cell type. Once you annotated your cell types, you can continue with your downstream analysis. In a single-cell toxic experiment, however, the output is the accessibility of the genomic regions. And unlike having genes, we do not have cell type specific genomic regions that can be used for cell type annotation out of the box. In this project, we investigate if we can use sorted bulk toxic data for cell type annotation of single-cell toxic clusters. I will start with introducing our data. We started with primary blood data. As for the single-cell toxic, we are using 10x genomics single-cell multiome data, but we are decoupling the measurements and only focusing on the single-cell toxic part. We have two bulk toxic data in differing granularities. First one is titled as human immune cells, and this dataset consists of 25 different FACS-sorted human immune cell populations, shown here in the lineage tree. This is our higher granularity dataset. The second dataset is the human hematopoietic differentiation dataset. This consists of 13 FACS-sorted primary cell types obtained during differentiation process. Due to time limitations, I will be presenting the results from human immune cells, but please feel free to check out the results from the differentiations using poster. As you can imagine, this single-cell and bulk toxic data needs a common feature space to be represented and to be compared. We decided to use ENCODE candidate cis-regulatory elements, scRNA-seq in short. scRNA-seq covers almost 8% of the genome, and they are derived from almost 10,000 experiments from thousands of samples. Therefore, we believe that scRNA-seq provides a good representation of regulatory genome. Here is our workflow. It consists of 4 different steps. I will walk you through the steps and show you some of our results. The first step is clustering. We start with single-cell toxic data, encode scRNA by cell matrix, apply some sort of filtering and pre-processing, and perform low-1 clustering. We visualize the results in UMAP space, and here's how the single-cell toxic PBMC data clusters looks like. It resulted in 20 different clusters, some of which are kind of separated from the others, some of them are intermingled. The next step is pseudobulk generation. Single-cell toxic data is too sparse to be annotated in individual single-cell level. Therefore, we need some sort of aggregation. And we use pseudobulk generation as an aggregation scheme. What we do is relatively simple. We take each an individual cluster and aggregate the cells within a cluster and create what we call pseudobulks. As we had 20 different clusters in PBMC data, we now have 20 different pseudobulks. Now the question is, how can we annotate these pseudobulks? As I mentioned earlier, we aim to leverage FACS-sorted bulk toxic data for cell type annotation. How we do this is relatively simple. We train a PCA space with the single-cell pseudobulks and project the bulk toxic data onto the same space. The idea is, as in any PCA plot, the closer the points are, the more similar they are. Here is our projection result. I'm showing you 3D PCA plot where the triangles represent pseudobulks and every dot represents bulk toxic data. Using the same logic, we can say that cluster 5 is a B cell and cluster 18 is a dendritic cell. However, it's harder to delineate the relationships here in the left corner. On the other hand, this is mostly a biological problem because our bulk toxic data includes multiple T cell populations. This causes overprojection of T cell populations onto each other. Overall, as you can see, using projection approach, we were able to segregate and annotate our single-cell toxic pseudobulks. The next step in the workflow is transcription factor footprinting. We couple our analysis with transcription factor footprinting with the idea that similar cell types show similar transcription factor profiles. Therefore, we applied footprinting for both bulk ATAC-seq data as well as the pseudobulks. We then train a classifier using the bulk ATAC-seq footprinting results and predict the cell type annotation of the pseudobulks using the classifier. Here I'm showing you the previous projection. This time the rectangles here represents the cell type annotations obtained from the classifier. As you can see here, we can confirm that cluster 5 is indeed a B cell and cluster 18 is indeed a dendritic cell. Moreover, we can also delineate more of the relationships of the T cell populations using transcription factor footprinting based classifier. With that, I'm already in my summary. I hope I could convince you that ENCODE CRES arrays provide a precise feature space for cell type characterization. Also, our projection approach coupled with footprinting successfully annotates the cell types in single cell ATAC-seq data. So this brings me to the end. I would like to thank my supervisor Martin Wingron for his support. UFA helped me a lot with the footprinting analysis, and I would like to also acknowledge my Duke collaborators. Here at the bottom you can see my contact details. Please feel free to reach out if you have any questions and if you would like to simply chat. And I'm looking forward to hearing from you. Thank you for your attention.